when do you know what's really kind of going on?
Yeah. Yeah, so I mean, I mean, I got confused before because I was looking at this like, oh, this is already a high energy state, so what are we doing turning on pyruvate kinase? The way to look at this slide is to say, well, there's a lot of glucose coming in, what's going to happen to it? So you can run this pentose phosphate pathway, which is basically this, right? Or you can run a lot of glycolysis. And what this is basically saying is that if you run a lot of pentose phosphate, it helps you run a lot of glycolysis because it turns on the gene to make an enzyme that's important for glycolysis, right? The pyruvate kinase. And so the idea would be, well, here we're still in the low energy state. So for any problem, I would have to tell you, have to be clear. Is it high energy? Is it low energy? If we just have a lot of glucose, I'm not telling you about the energy state. What can happen? Where can glucose go? Well, there's a number of places it can go. You know where they can go, where it can go. Yeah?
Just a little bit curious about that time scale. Because doing this and going through transcription and everything is going to take a lot longer than just sort of the right ways of regulation.
That's true. That is true.
So does the body have a way of knowing that there's going to be a lot of glucose and actually making those enzymes is going to be a while?
No. I mean, I don't think the body can predict the future in terms of the glucose level, but
I guess another
question. It's only going to make as much messenger RNA as glucose continues to stimulate. So it'll make a little bit, and if the glucose stops, it'll stop making it because this pathway will no longer be active. So it will be proportional in the sense that you can make some glucose, and then so that there'll be a time lag, so then you can make more ATP from that glucose. But in the meantime, if you got shifted into a condition where you really need to go the other way, then the other signals will be counterbalancing. So, we're just kind of showing one slide at a time for like what can happen. In reality, everything is happening. Everything is always there and available to happen. If the signals go the other way, this may get swamped out, right? You could be right. I mean, it could be that before pyruvate kinase gets made off the ribosome and is ready to work, something else has already happened. There's no more glucose, and moreover, we need to run gluconeogenesis.
What's the approximate time scale?
At least a few minutes. It depends on the cell type, though, too. Right? I mean, here I guess we're pretty much looking at a eukaryotic cell belonging to the nucleus. So there are parallel processes in bacteria, but they don't require the transport.
So it's at least fast enough if you're eating a meal.
Yeah.
To process all that glucose, this would still be working like on that same.
Oh yeah. I mean, in a matter of minutes, it can work in. Yeah. But I've also talked about minutes as being kind of a long time because if we're thinking in the context of bacterial cell division, E. coli doubling time is like 20 minutes. Mm-hmm. So if it takes you 2 or 3 minutes to turn a gene on, that's a significant fraction of your total life cycle before it's time to replicate again. So it depends on the cell and the context, what is a long time. Here, this would be, I guess, still relatively short.
How does the body adjust to large fluctuations in these substrates? For example, you're having a meal, you're eating stuff, and then you have another meal really quickly that's really high energy. The system's very kind of slowed down and it's going somewhere else.
Well, I mean, that's what we're trying to figure out here, right? That's what we're trying to, we're getting a lot of shocks to the system. Tons of glucose is coming in. One way the body's adjusting is by turning it into fat. Right? Another way that it will adjust is by making glycogen out of it.
But you can't just stop it halfway through.
You mean like block the glucose from coming in at all? Well, there is an equilibration mechanism too, right? There's the transporter mechanism, and then there's the activity of hexokinase. So there's lots of control mechanisms. I mean, the carbohydrate metabolism and this energy state is like probably the most heavily regulated process in the cell. So there's layer upon layer of control. So, here is, for example, insulin, right? High blood sugar comes in, and it binds insulin receptors. This is stuff that you looked at last term, I know, but just as a kind of review in a sense of how pleiotropic insulin's effects are. You know, these are all the enzymes that insulin upregulates. These are enzymes that insulin downregulates. These are gluconeogenesis enzymes. Right? Gluconeogenesis enzymes are downregulated by insulin because they would make more glucose. You definitely don't want these enzymes activated by insulin. There's already a lot of glucose around. So the ones that get activated by insulin are the ones that break down glucose, or that take the breakdown products of glucose and then make something else out of them, like fatty acids. Okay. So, you know, there's an insulin receptor. It gets autophosphorylated in response to insulin. Insulin doesn't actually get into the cell, right? But it triggers conformational changes in the receptors, which ultimately trigger phosphorylation of a protein called IRS1, which operates through a set of other interactions to ultimately phosphorylate a protein called RAF1. Activated RAF1 phosphorylates a protein called NDK, which phosphorylates ERK, which moves into the nuclease, which phosphorylates ELK, which allows ELK to bind SRF so that that can bind DNA and you can make new proteins. So there's this whole cascade, right? And so you can amplify the signal by virtue of this cascade, because you can go off and hit more and more molecules. Or you can have different levels of control on the cascade, so that, for example, if you need 2 phosphorylations on ERK, but maybe a third one would be better, you know, it's not necessarily just 2, or 1 or 2 or 3 or more. More of it can get into the nucleus or not, right? And how much? How many, you know, how much of this protein is around? What is the saturation on the DNA of that protein? How much gets made of the new proteins? So you can have very fine levels of control if you take it through a complicated pathway. And you can use this pathway to kind of branch off in different directions, which is something we'll see on another slide. You know, here's another aspect of it. Here's insulin coming in. We want to turn off gluconeogenesis now because it's insulin, so we don't want to be making more glucose in the cell. It goes through to PKB, the last slide we've shown. PKB is a kinase, so it's going to phosphorylate something. It's called phosphorylate this FOXO1 protein, which is something called a fork-head transcriptional activator. And so if that's phosphorylated, however, it gets recognized by ubiquitin, which is a system to break proteins down. So phosphorylation breaks this protein down. If it's broken down, it can't get into the nucleus, and therefore it cannot turn on gluconeogenesis enzymes. So this is basically a way for insulin to work by blocking a protein that would normally activate gluconeogenesis enzymes and effectively destroy it by phosphorylating it and then having it become a target for breakdown. So there's lots and lots of really ingenious mechanisms by which the cell operates to keep things on or off. Right? So this is just showing you a different one. And the levels of control on the DNA, right? So we just see that we just saw 6 or 7 or 8 steps of kinases, layered kinases from insulin into the nucleus to phosphorylate some transcription factor. Right? That would be FOX01 here, this fork-head thing here and here. Right? But what about all these other guys? Right? I mean, this is a highly fanciful drawing, but there actually are genes with all these crazy letters: DAF2, GRE, TRE. Who knows? There's a whole bunch of stuff. 1,500 nucleotides upstream of some synthesis. Here's polymerase ready to synthesize a message out here, and here's something like 20 different factors. Okay? So think about how complicated this is, and recognize also, and get into this more next term, it's not just a linear piece of DNA, right? This DNA is supercoiled in the nucleus. It's curved up on itself. It's a tertiary structure. These proteins are binding all over it, to the DNA, to each other, making this very complicated tertiary organization. Right? We don't understand it in any detail, but we certainly know it's not a linear piece with all these proteins lined up on it. Right? This is just art, not reality. Okay, questions?
I'm struggling with the amount of detail in the book and even in some of the slides, like looking at this whole pathway of insulin, and like, to what level do you want us to know this stuff from memory, being able to draw out these pathways? Like,
You certainly don't need to know stuff like this from memory.